Okay. And we're on.
Okay, so we have Deborah Rodrigues with us today. She has her BS and MS in biology and microbiology from the University of Sao Paulo, Brazil, and her PhD in microbiology and molecular genetics from Michigan State University. She then did a postdoc at Yale University, and she's now assistant professor at the University of Houston in the Department of Civil and Environmental Engineering. It's a pleasure to have you today.
Thank you. Thank you for the nice introduction. Thank you very much. Well, today I'm going to talk about my research that I've been doing the past 3 years. Well, it's not completely the whole thing, but I want to give you an idea about, because there is as much as I can talk in 1 hour, right? So I had to actually give you some highlights about what I've been doing. Like Amy was saying, I'm an environmental microbiologist, and but I have also been working with nanomaterials and the fate of nanomaterials in the environment. I'm very interested in emerging contaminants in the environment and how it affects microbial populations and the environment. And I'm going to explain why. But first, today I'm going to be talking about carbon-based nanomaterials. It's right from here. That's not going to do. Anyhow, okay. Well, just to introduce a little bit, what are nanomaterials? Well, nanomaterials is anything between complete systems or materials that are between 1 and 100 nanometers. I put this figure here just to give you an idea about the size. Here you have a hair. This is a nanofiber. A hair is very, very small already, but a nanofiber is way smaller. And here you have the nano size here. You have a water molecule and even a virus, and here are some nanomaterials that you can see. It's very small, almost the size of a water molecule. So nanomaterials are very, very small types of materials. And they're already being used in the market for different applications. From health products, some of you use sunscreen. Some of them have nanomaterials on it, also paints to prevent mold growth. But some toys and silverware, and even in your cars, you can have also nanomaterials. So it's already being used in the market. And the ones that I'm interested in are actually the ones related to carbon-based nanomaterials. This is stronger. Okay. And most of the carbon-based nanomaterials, they are more in electronics and light materials. And just to give an introduction about what a carbon-based nanomaterial are, they are carbon molecules, basically. They are really carbon. And they have different shapes. They can look like a flat sheet, which would be called graphene. Or you can have modification of these graphene sheets here with COH groups, OH groups, and other functional groups, and those are called graphene oxide. You can have these graphene sheets instead of like a flat paper, like rolled, like a tube, and those are called nanotubes. If you have just one tube, it's a single wall nanotube. If you have more than one, then they are called, of course, multi-wall carbon nanotubes. There are other types, but those are the ones that I'm actually interested in and I've been investigating throughout the years. And why do I care about these materials? Well, these materials, they have exceptional strength, a high thermal conductivity, and high surface area. Therefore, they can be used to a lot of different applications in biomedical, biotechnology, optics, fuel cells, energy storage. So there are so many applications, and eventually they will end up in the environment. That's my concern. And we know already, there are a few studies that have been shown that there are potential serious health and environmental impacts for these materials. This is still a study ongoing, but these preliminary studies were kind of scary because showing some preliminary health issues because of their high surface area, because they're very, very small, so they can penetrate cells more easily and penetrate microorganisms. And this can actually be persistent because they are not usually biodegradable. At least people don't know yet if it is. So we are trying to understand that. So therefore, the objective of my talk today is really to understand how these nanomaterials are toxic to microbial population. As a microbial ecologist, I'm really interested in what they do to the microorganisms and how microbes react in the presence of these nanomaterials, basically. And understanding that, we can also develop new applications for environmental engineers. If we understand these materials, what they do in the environment, how we can modify them or make it better or safer for applications in environmental engineering. That's the main idea about using. So the outline of my talk, I'm going to start to explain the nanomaterials, how they affect microbial cells, and what are the mechanisms of their toxicity to microorganisms. This is going to be with pure cultures in the lab, but then the lab doesn't show really what happens in the environment. So the next question will be like, okay, in the lab, this is what happens. What about in the environment? Would still the environment be affected by these nanomaterials or not? And how they would be affected? And the next question would be how we can safely use these carbon-based nanomaterials. What modifications can we make to actually make it safe for use if they are toxic? And I will briefly talk about other research interests and other projects that I've been doing in my lab as well, to give an idea about what's going on in my research. I'm going to start with the carbon-based nanomaterials and discuss the toxicity of microbial cells and their mechanism of toxicity, how they are toxic to the cells. Like for us, we know that if we get a cold, we have this virus that enters us, we start to sneeze, right? What would happen to the microbes? What would be their reaction in front of these nanomaterials? That's what we want to know, basically. And for that, we are using two model organisms, E. coli and Bacillus subtilis. Why that? Bacteria has two main groups. They have the gram-positives and the gram-negatives. Oh my God, what is that? Okay, I'm going to explain. Follow with me. So the gram-positive, which is here represented by Bacillus subtilis, has a large peptidoglycan. It's a cell wall that is very thick compared to the gram-negative that has very thin one, but it also has a membrane. This one doesn't have a membrane. So they are different, and because they are different, their cell structure is different, they will behave differently with contaminants, and they will have different response. They are not the same, so they cannot react the same way. That's the idea. And we pick those two microbes because they are found in the environment very frequently. And more importantly, since I'm using some molecular biology tools, some biotechnology studies, I need to know their genomes as well, and they have been sequenced so I can actually understand at the molecular level what's going on with this microorganism. I promise it's not going to be very hard. You guys are going to be able to follow it. And if you don't, please stop me, I don't mind. So, the first thing that we analyzed was like, okay, we got this microorganism. Is this nanomaterial toxic to this microorganism? So we got a sample with the nanomaterials and we exposed to the bacteria for about 1 hour. And then what we did, we stained with two different dyes. And I'm going to explain, I like to use my hands as a Latin American type of, I like a lot to use my hands. So if you have the cell that is like a shell, like that, so if there is damage of the cell wall, the dye will get in. And the dye that will get in is the propylyl iodide, the red one. And it's going to stain the cells in red. Now, if there is no damage in the cell is intact. There is just one dye that can penetrate the membranes. And this one is the green one. So the green one, we stain all the cells that are intact. And the red one will penetrate only the cells that have been damaged somehow, which we don't know how, but we're going to go there as well. And here's the result. Remember in the beginning I told you that I was interested in the graphene, that is a flat sheet, the graphene oxide that has the functional groups, the COH, OH, and also the single wall nanotubes that is just a tube, and the multi-wall that has several tubes. And then I have my control that didn't have any nanomaterial. And then I look how many cells were getting inactivated, meaning killed, that are not alive. So we compared the different nanomaterials with E. coli and bacillus, we look at how many of them were inactivated, and you can see that at the single-wall carbon nanotubes, the higher the number, more cells were killed, basically. So the single-wall carbon nanotubes had the most killing. Of them, you can see that there was some inactivation. They were kind of inactivating the cells at some extent, but at different extent. Graphene was the second one. Graphene oxide was a little bit less than graphene, and multi-wall carbon nanotube a little bit less. So we decided to investigate how, since those two here, single wall and the multi-wall, were the highest and the lowest, we decided to investigate those two here to see what's going on with the cells. How are they being inactivated? We had the hint that it might be some cell wall being broken because of the dye that got in, but we don't know exactly how it's breaking the cells. What's happening to break the cells, right? So what we did, we used an experiment called DNA microarrays. Think about DNA microarrays as a molecular biology tool, but think as a sensor. It's a sensor. A sensor to detect the level of gene expression. Meaning, if the gene expression, the blue color or brown eyes is a gene expression that it's the RNA that is produced and produce the protein that generate the colors in your eyes or the color of your hair. And microorganisms, depending on if they are happy or sad or in a certain situation, they will have different types of genes being expressed to demonstrate that. Or when we are sick, we have our immune system that starts to produce a lot of proteins to fight against the microorganism that is inside our body, right? Microorganisms also have a mechanism of responding to that, and that's how we measure it. We measure through that, the level of RNA that is being expressed. And this sensor can detect that level of expression, the amount and which genes are being expressed. Is it clear? I know that sometimes can be a little bit confusing. Anyhow, so we tried different, we used the single-wall nanotubes, the multi-wall, and a control sample without any nanomaterial. We let them exposed to the nanomaterial to see how the cell is going to respond to that. The genes are going to be expressed. And we use this chip that has the whole genome of the microorganism. It's a tiny chip, but you can put the whole genome of the microorganism there to detect as a sensor, basically. And you can compare the single wall and the multi-wall with the control to see how different is the genes being expressed compared to the control. And the results of that is this, just a summary to give you an idea. So we had, when you compare single wall with the control, we have about 1,409 genes being expressed differently from the control. Meaning something, the microorganism is responding differently than the control that doesn't have any nanomaterial. What's going on? And then the multi-wall and the control had 424 genes. Less than the single wall, but it makes sense because the multi-wall was less toxic to the microorganism than the single wall, right? So there's probably something going on differently with those two nanomaterials. But when you compare both, you have 480 genes that are identical to the single wall and multi-wall. So there is an overlap on the response, and some response that are different only with the single wall. And based on these genes, it's a lot of genes, it's very complicated, very complex to analyze, but you can put together what's going on in the cell, basically. And one of the responses that we saw for both single wall and multi-wall was production of oxidative stress, meaning there are some reactive oxygen species that are being produced. What are oxygen species? Hydrogen peroxide, superoxide, and hydroxyl radicals are reactive oxygen species. And those are the types of species that makes us become old as well. The production of these is a way of deteriorating our body, but also it's a way of killing microbes as well. We use a lot of hydrogen peroxide to, when you have a wound, we put hydrogen peroxide to prevent microbes to infect an area, and that kills microbes. That's the reason why. And guess what? Newsflash. This hydrogen peroxide is also killing the microbes somehow because they are being produced by the carbon nanotube. And we know that because we saw several genes related to oxidative stress, like the catalase gene that try to remove this hydrogen peroxide, and the superoxide is mutase. But there is as much that the microorganism can fight against if there is too much being produced. Everything in excess can kill you, and that's what is happening here. And you can see that this high super oxidative stress is damaging the DNA, is damaging the RNA, and it's also damaging the protein. And you have all these genes being expressed that show that this is happening for multi-wall and single wall. And here I'm not showing, but graphene and graphene oxide also produce the same type of mechanism as well. But then there is this other 1,000 of genes only in single wall that we don't know what's going on, right? That I showed in the previous slide. And this is related to cell damage and fatty acid recycling. Well, the membrane is full of lipids, which are fatty acids, basically. If you have damage, microorganisms, they eat each other. If one is dead, they are cannibals. Let's put it this way, in a simplified way. So if this nanomaterial is killing some microorganisms, what they're going to do, the ones that are alive, they're going to use these fatty acids that are in the media from the other bad microbes, and they're going to eat it. And that's exactly what's showing here. There is this fatty acid beta oxidation that it is the uptake of bad lipids in the medium that the microorganism is using to synthesize new membranes, either for repair or to grow, basically. And you also have the glycolysis that has glycolysis produce energy for the cell for survival. So we eat sugar, right? For energy. Glycolysis is part of it. And they also eat sugar to survive. And they need the sugar for energy to survive and fight against bad things in their environment. And that's exactly what they are doing here. So they have a lot of glycolysis to produce energy so they can fight against the nanomaterials that are trying to kill it, basically. So you have all these mechanisms that is happening in the cell, basically. Just to summarize that part here. So what's going on? Well, single wall nanotubes and graphene, from my initial data, we showed that it's the most toxic to the microorganisms. But in order for these nanomaterials to be really creating all this reactive oxygen species and damaging the membrane, you need direct contact with the cells. That's one thing that we showed. And there are two types of mechanisms that is causing stress in the cells. One of them is oxidative stress, that actually all carbon-based nanomaterials have shown to have that. And the other one that we saw with only CNT was cellular damage. I'm still under investigation for the graphene and graphene oxide to see if there are other mechanisms besides those two that we saw with single-wall carbon nanotube and the multi-wall carbon nanotubes. So this is going to be another story for later. Now, I have shown you that carbon-based nanomaterials, they are toxic and how they are toxic to microorganisms. Now, the question is if it goes to the environment, because we're producing all these products with nanomaterials, if they go to the environment, what's going to happen? Can it damage the environment? Because in the environment, you have a lot of chemistry involved as well. You have salts in the water, you have organic matter, you have all these things, especially wastewater. That could be one way that this nanomaterial could enter the environment, or even landfills. I've been working both ways, but today I'm going to just present the wastewater effect, if it would affect the wastewater treatment, basically. So, microorganisms, well, they are harmful, but just 1% is harmful. The majority, we couldn't live without them. I don't know if I'm breaking any news to you, but I like to tell my students when they take my micro class, they are good? Yes, they are. They are very good, and they help us a lot. Without them, we wouldn't have this beautiful place, because they clean. They clean the waste, they degrade all the waste that we generate. All the wastewater treatment plants, they are biologically based, and you need them. So, imagine if we put nanomaterials, what would happen? And we kill microbes. Well, my question is, microorganisms, they are responsible for example, removal of nitrogen and phosphorus in the wastewater. And if it kills those microbes, and then we throw the water in the rivers, well, we're not going to have this beautiful picture, but probably something like that, disgusting, right? And that's why I want to actually investigate what would happen if these nanomaterials went to the environment. That's my hypothesis. If we would have disruption of this cycle, degradation cycle, we would probably get some degraded environment as well. So my approach, actually my group's approach, because my students were the ones doing the work, we got some water sample, some sludge from the aeration basin from the wastewater treatment plant in Houston. And we did several tests, but I don't have time to present everything, so I'm just going to say a few ones. So we look at the concentration of graphene oxide that would actually affect the microbial community in the wastewater. So we test different concentration. We use graphene oxide because graphene oxide disperses very well in water, better than the other nanomaterials. Graphene oxide, because of all the functional groups, if you put in water, they look very well dispersed, and it's nice to work with. And we look at the effect of the graphene oxide in the biogeochemical cycle, the carbon, the nitrogen, and the phosphorus, to see if they were affecting those cycles. And the first assay, we were just analyzing the concentration, and we look at the heterotrophic plate count method, that is looking at the whole population, how the whole population of wastewater is actually reacting to the presence of these nanomaterials. And then we look at the effect on the carbon, nitrogen, and phosphorus removal. The carbon degradation we did through the BOD test, and the nitrogen and the phosphorus we use the colorimetric method. But I'm going to talk first the BOD, the plate count. So we got the activated sludge, we tried different concentrations, and then we played them using the standard methods technique. This is actually the food for the microorganisms. You need to give them food in order for them to grow. And each colony here, each bubble here, it's a cell of bacteria there that is growing. And you can count them. And you can determine how many microorganisms how much is reducing with the presence of different concentrations. So we use different concentrations of GO from 0 to 300, and we analyze how much of the microorganisms were actually not growing, basically. And we saw that from 50 milligrams per liter, we start to see some effect on the microbial community. So this concentration, okay, 15, it's quite high, but still possible to have if you're mass producing nanomaterials. Oops. Okay. So the next question was like, okay, we saw that it's affecting the heterotrophic plate counts, the majority of the community. But what about the specific functionality of the microbes, like the carbon, the nitrogen, and the phosphorus? Because the wastewater, we aim to eliminate those before we throw in the rivers, right? We don't want that. Otherwise, we have all this heterotrophic growth and the other picture that I showed you. So we did the carbon degradation. We used the activated sludge. We put the different concentration of GO. We measure before adding the material. Time 0, and then after 5 days of exposure. And we look at the BOD. And we saw that as low, here's the concentration of graphene oxide, and here is the BOD. And we saw that at 10 milligrams per liter, it starts to actually affect the community. I mean, it's not degrading the organic matter as it should at 10 milligrams. So it seems like that this community, specifically the carbon one, the ones that are responsible for removing the organic matter, is actually being affected at lower concentration than the heterotrophic. The second one was the nitrogen and the phosphorus removal. We used the colorimetric method. For the nitrification, if the community is working nice and well, what you're going to see, you're going to see the whole degradation happening, the microorganisms happy. So the ammonia is going to go away, and you're going to have nitrate. I'm just looking at the aerobic part, nitrate being produced. For the phosphorus, there are some bacteria that they remove from the wastewater by accumulating inside their cells, the phosphorus. And those are accumulating phosphorus bacteria. And if this microorganism is not active, then you are gonna have a lot of phosphate in the water, basically. So it's not gonna disappear from the wastewater. So what we did, we measured again the different concentrations for the nitrogen. And what we saw, remember, we are adding ammonia, and the ammonia should disappear over time in the wastewater, right? To produce nitrate. But what we saw is that the ammonia here is actually increasing over time, and the nitrate, instead of increasing over time, because it's been degraded, is actually reducing. So it's not being produced. The nitrate is not being produced, and the ammonia is not being degraded. So the community is being kind of inhibited. And we start to see a significant value at 50 milligrams per liter, similar to the heterotrophic weight counts that we saw earlier. For the phosphorus, it seems a little bit less sensitive because we saw only at 200 milligrams per liter a more significant inhibition of the removal of phosphorus that also was accumulating instead of disappearing over time as it was supposed to. So in other words, graphene oxide, depending on the concentration, can affect the different functionalities in the wastewater. So it is a concern to just throw it away as it is in the wastewater. And we might be harming the wastewater treatment plant if we don't watch out for how much we are putting there. So, but nanomaterials are fascinating. They can do so many good things. All the electronics and everything, we don't want to get rid of nanomaterials, right? We want to continue to use them. So, how can we safely use these carbon-based nanomaterials? Are there ways that we can do that? Well, some people have come up with different solutions for that by reducing the amount of nanomaterials by combining with polymers. So you make polymers and you reduce the amount. Like you have the touch screen here, that is a PET polymer combined with graphene. And it's very flexible, touch screen, solar cells, electronics, and even paints are all combined with polymers. And when you add the polymer, there is another benefit to it. The polymer have also properties, and you can combine the properties of the nanomaterial with the polymer. For example, by combining this one, you can increase the contact angle, meaning you have less microorganisms attaching to it, and less stuff attaching to the ship hull as well that would reduce friction, right? And it would less energy for the boat and everything. And you have some other polymers that they can increase also the antimicrobial properties. And my research group is interested in the polyvinyl-N-carbazole. Why is that? Well, the polyvinyl-N-carbazole is this molecule here, and it has aromatic rings, like the carbon-based nanomaterials. They also have aromatic. So they can form pi-pi interaction, meaning you can intercalate the graphene with the polymer, graphene with polymer, basically. And because you can do that, you get a better dispersion of this material. But on top of it, we were able to reduce the concentration by only 3%. So we had more polymer and less nanomaterial. And we still have something very stable over 30 days. For GO, it's not so much a problem, but for CNT, it's very hard to disperse them in solution. They tend to aggregate. And with this polymer, since it's from the pi-pi, you can disperse and you can keep them for a long time, very well dispersed for different applications. But the first thing that we had, the question that we had, okay, would that be toxic to humans if you add this polymer? Because I want to use for environmental applications. Drinking water, maybe coating pipelines for water, things like that. So the question is, if it goes in the system, am I going to be sick for that, or can I survive? So we did the human toxicity assay with the polyvinyl-N-carbazole, PVK, with graphene oxide, and with single-wall carbon nanotubes. Single-wall carbon nanotubes are extremely toxic to humans, has been shown already, but I'm going to show the data anyway. So we grow the human cells, we incubate them, let them very happy and all, and then we put the nanomaterials to see what the nanomaterial does to the cell. And in this plate you can see the darker the color, or the lighter it goes, means the cells are less happy, meaning they're dying. They're not very happy, so it's kind of concentrated dependent. And then you can measure that change in color, basically. And you get the following result. So here is the cytotoxicity, meaning how many cells are being killed. So 100% meaning a lot of cells is dying. And you have here all the graphene oxide. So graphene oxide, the concentration that you used wasn't very toxic to humans. But with the PVK, since we are adding just 3% of graphene oxide, we reduce almost to zero the toxicity. And then we have the control with dead cells as well. And the single-wall carbon nanotubes for human cells is pretty high, naturally. And with PVK, we were able to reduce a lot the toxicity with that, which is actually a very good thing. So, yes?
Are you plotting the toxicity or the color that you mentioned?
No, we measure by toxicity. We have a control. Yeah, we measure the color, but then we convert to toxicity levels.
So is the conversion a well-understood process? Or?
Yes. Yes. It's a standard method. It's actually a standard method for.
So the error bar you have on those plots, is it error bar on the color or on the inferred toxicity?
No, the inferred toxicity. So now that we saw that we were able to reduce the toxicity of the nanomaterial to humans, can we use for, let's say, environmental engineering applications like modifying membrane filters? It's more like a. And so the idea is, since we reduce the concentration to 3%, would we still have the same antimicrobial properties of this material or not? I mean, we reduced the toxicity for human cells, but did we reduce the toxicity for microorganisms? Can we still use for water purification, for example, water treatment? So we prepare our mixture with 3% graphene oxide, and we prepare our membranes by coating them. And then we did a series of tests, but I don't have time to go through all of them, so I'm going to present 2 tests that we did that gives you the overall idea. We test on the surface to see how the microorganisms were surviving in the surface. Whatever is retained in the filter, are they dead or alive, basically? And this is the water that people would drink if you still have microbes or not, and how many microbes you have there. It would be safe to drink the water. So we did the first assay, that was the live and dead assay, the same, similar to the initial one that I mentioned. So the cyto9 penetrates all cells, and the PI, the propylium iodine, penetrates the ones that are damaged, basically. They are membrane damaged. And we look in the surface after putting different microorganisms filtering through. And we saw, here is the percentage of cells that were inactivated. We saw that the GO filter had much more GO than this one. And we saw that we were able actually to produce similar results of cells inactivated of E. coli in the presence of the new nanocomposite. And for bacillus, similar result. Actually, the PVK GO did a better job actually than just pure GO. So we actually got a good result that could be used for water treatment. And we reduced the trash that we are going to put in the wastewater, basically. Now, can we drink the water here? Because we filter, I mean, you killed there, but what about here? How much did we actually remove? So we got this solution here, and then we gave food to the microbes, and we were able to count how many were alive, basically. And when we look at the log removal, how many cells, based on the control samples, we saw that with the PVK-GO sample only, for Bacillus and E. coli, we had about 3.5, almost 4 log removal of both microorganisms, which is pretty good. And for GO samples, it's slightly less, but still very good. But the GO, but here you have only 3%, here you have a bunch of GO. Just keep that in mind. So you're using less material, nanomaterial. So as a conclusion, we saw that it's possible to embed the graphene oxide in a polymer and still have antimicrobial properties that could be applied for environmental engineering applications. And we included only 3% of the nanomaterial, so we are reducing the waste that we are generating of this nanomaterial to the environment, which is a great thing. So it could be also a very economical option because nanomaterials can also be expensive, so it's a good deal, reducing the concentration and everything. Now, I want to briefly talk about my future research interests and future directions. I talk mostly about the nanomaterials and nanotechnology applications for water treatment and all. But my interest is really combining both technology. My research group has been studying also microorganisms, how we can use microorganisms to treat water and other applications. My research is focused on combining those two for water treatment, but also reducing of carbon emissions and energy production. I'm interested in these three things, actually. Ambitious, I know. But I mean, this is part of the NI Grand Challenges, and it's very timely, and the idea is really to use microorganisms and also nanotechnology, but in a safe way, trying to understand the fate and the toxicological aspect of nanotechnology, because it's still a new thing and we don't know what's going to happen if we just dispose in the environment. We're still learning the effects to the microbes and what's going on. And of course also how this nanotechnology can and biotechnology can help in the emerging contaminants such as pharmaceuticals, estrogens in the wastewater. There are a lot of things that are coming up that microorganisms, they don't necessarily know how to handle it, even though they can handle a lot of stuff, but there are new stuff that they might be able to handle in the future. But maybe if we combine both technologies, microorganisms and nanotechnology, we can have a better cleanup and remove of pollutants from the environment. And at the same time, I'm interested in using different microorganisms, not only bacteria, but also working with algae as well, because algae is a very good microorganism that can sequester CO2 and can also produce energy, like biofuels. And of course, to be able to work with microorganisms, we need to understand how diverse they are, what their functions, what they are doing in the environment, and how we can use them for these three things, actually. And just to give an idea about one project that I've been trying to combine nanotechnology with biotechnology is really using, this project started recently, and we are combining, it's a molysulfide, but we are trying other nanomaterials, that this material can produce water splitting, so it produces hydrogen, which is a very clean energy, no CO2 production, which is great, and at the same time, you can inactivate microorganisms as well. So you get clean water and clean energy at the same time. That's where you are actually combining nanotechnology and a little bit of biotechnology by removing the microorganisms as well that you are interested in removing. Another project that my research group is working on is with algae. We have been trying to understand different species of algae and see how they can actually produce lipids, what kind of lipids they are producing. We are mainly looking into lipids that are related to energy production, but we are also looking at different applications because algae is very important for pharmaceuticals and omega-3 production and different products that you can use for diverse types of industries, so I'm not limiting myself to energy production. And we are using different technologies to try to understand this microorganism, and because most of the studies have been done with specific microorganisms and focusing one, I'm trying to understand really the community, how they are working, how they can benefit for energy production and even water treatment as well. Here I'm talking about wastewater, but I'm also talking about manure treatment and different type of wastewater. I'm not only interested in one type, and see how these different microorganisms work. Now, the third one, which is more in the civil side, I want to make everybody happy, so I didn't forget the civil folks. This project actually started with a colleague. Well, she works with concrete and self-healing concrete using fibers and different types of self-healing materials. And then I told her, like, can't we use microbes to do that? As a microbiologist, I was like, yeah, they can do it as well. How? Well, microorganisms, in some places, more alkaline places, like caves or alkaline ponds. This is a Turkey picture. My student that is working this project, she's from Turkey, so she got a sample from Turkey from an alkaline pond there. And we are trying to investigate how these microorganisms, they can produce calcium carbonates to seal self-heal cracks in the... A lot of studies that have been done with that. There are some folks in Delft that are working with that. But they use mostly sporosarcine, one type of microorganism. I'm trying to understand the community and if there are anything that is better than what they are using right now, and how they do, and what type of calcium carbonate, how they precipitate it, and what's going on. And suddenly I came across, hey, to precipitate this, you need CO2 as well, and you need calcium. So I ended up going also in the direction, okay, so I can do calcium precipitation and at the same time sequester CO2 as well. So do calcium carbonate precipitation with CO2 sequestration. So we have been investigating how these microorganisms are actually sequestering CO2 and at the same time self-healing cracks. And there are other lines of projects that we are thinking of pursuing as well. All these tools, all these projects that I've been working on, I use different tools, traditional environmental microbiology tools, but also molecular biology techniques. I mentioned a few, but the idea is really to understand how microbes look like and what's the interaction of microbes with pollutants in the environment. So for that I use a lot of different types of microscopy, and I can see how microbes, is the carbon nanotube really puncturing the cell, so you can see that. Or you can actually see the shape of the microbes. I show you the picture of the algae, the different shapes, so you can actually also identify some microbes as well. Another technique that we use is, well, some bacteria. They look like little dots in the microscope. So how can you identify them, right? They look all the same. So the idea is to actually use different techniques that it's at the molecular level to separate them, distinguish them, and also to understand what they are doing in the environment, what their metabolism, what can they do. And again, the molecular biology techniques are very powerful to measure exactly what they are doing, and how they behave, how they answer to changes in the environment, and how they react to the presence of pollutants, emerging pollutants in the environment. And so these are different toolkits that I've been using to answer all these diverse questions that I presented today. Of course, as an educator, I'm engaged also in outreach programs with different students. I have a program with the research experience for teachers from NSF, and every summer I host high school teachers that they come to my lab, and we work together on different projects that they can take to their classrooms and prepare teaching modules to present to their students. I also love to have high school students, and I always encourage them to participate in science fairs. And I was lucky because I've had a lot of very brilliant high school students. One of them ended up at Yale, another one at UT Austin, another one is doing pre-med at Baylor. So, and this one is an example of one student that she won 2 prizes in a science fair in Houston. One for the environmental side, and one for the whole science fair of the project that she worked related to the filter that I presented to you today. And I don't forget my graduate students either. Of course not. My graduate students, I always try to get them fellowships and prestigious stuff. This student here, she is actually a Hispanic American. We got her a scholarship to spend a year in Brazil, even though she doesn't speak Portuguese, but she's learning. But, well, she speaks Spanish, so it's not too bad. She could make. So she's spending a year, she got the Rotary scholarship. This is the Congressman Ted Pope that is giving her the Rotary scholarship. And I have many other stories with my graduate students, and even undergraduate, I always try to get them scholarships and awards and things like that, nominate them for awards and things like that. And but I don't have time to say everything, so I'm gonna stop here now. And I'd like, of course, thank my research group. I showed this picture once, and people ask me, like, "You have that many students?" I was like, "Well, I have undergraduates in this picture as well." He's an undergraduate, she's an undergraduate, and I have some visiting scholars. Those two are visiting scholars. He already graduated, but she was a postdoc, she left as well. But yeah, most of my students that did the work, they are PhD students, and some of them, they have their own fellowships, some of them are from my funded projects. And I would like to acknowledge also my funding sources, my NSF Career Award, the NSF Corp, and the UH Early Faculty Grant that I got, and of course my collaborators. Some from Brazil that are hosting my student in Brazil, from University of São Paulo, and from Institute of Research in Nuclear Energy, because this colleague is also interested in removing radionucleotides. And I told her, like, I'm not going to work with radioactive material in my lab, but if you want, you can test the filters, you can investigate that there. So she's actually doing some work on that. And a colleague from Case Western University, I work with him, he's a chemist, with the development of the filters, because I'm not a chemist, and although I learned a lot about chemistry, but I'm interested more in the microbial processes, and so I've been doing a lot of collaboration with and that's all that I have, and I'll take any questions. Yes? Thank you.
You sort of talk about nanotech being in a Wild West phase. We don't know what they do. So what have you identified in terms of health impacts or environmental impacts that are actually happening out there because people are using them in ignorance of the stuff you're researching?
We don't know yet what's happening outside there. And this is one question, even the EPA, they haven't put any regulations about nanomaterials because they don't know what it does. And there is a problem with nanomaterials. There are so many different types. And each one have different impacts. There is several groups that are working, there are some centers of nanotechnology that are working with silver nanoparticles because it's the most widely used right now. And they are investigating and trying to figure out what's going on. But carbon-based nanomaterials, I mean, here in US, I know, I talked to Barbara Kahn from NSF, where I got my project funded. She told me that there is just me and another guy, another professor from Brown University that is working with this nanomaterial. So there are not many people, at least in US, working with that, and the toxicological aspect of it is still like starting. We're starting to know, but we don't actually know the impact yet.
And there's no pain point out there that drove them to give you the money to fund this research and so forth?
Yeah, there were some preliminary studies that were given by Pedro Alvarez and even Manny's group. And then we have the nano health part of NSF that is related to affecting the environment. But the reason of this research is because people don't, they know that there are some potential toxicity effect, but the majority of the studies were done in the lab, not really in the environment. And my research group has been focusing on going to the environment and see what's going on for carbon-based nanomaterials. Some other groups are looking at other nanomaterials in the environment as well, like silver nanoparticle is the most studied, actually, and it's the most used. But we don't have any regulations. The idea is really to see if we can develop some regulations for that at some point. So, yeah.
Isn't there a center at UCLA? I attended
Yes.
the presentation that they do a lot of work on environmental impact of
They work more with silver nanomaterials. Yes, and UCLA with Duke University.
That's
Yeah,
right.
yeah.
And I have a follow-up question. That was an excellent presentation. I attended a meeting a few years ago. The national security implications of the nanomaterial, particularly as used as a dirty weapon or dirty bomb, and the issue of the public health issues. I remember going back to the previous question, I read a policy paper that they said that for the plants that they are producing this nanomaterial in the nanotubes and others for the workers' health and safety, I remember reading that line, and I want to see what is your reaction to that. They said the filters that they use for the face masks of these workers when they are producing nanomaterial is as if you use a fishing net to catch mosquitoes, meaning that they are very ineffective.
Yeah,
What's going on in that domain, in occupational health and safety area, for the workers that they work at the plants that they produce nanomaterials?
Well, the same way that we don't have regulations, there was a debate with some folks from the NSF of how our students are using these materials and work in the lab. I always make my students work in the hood.
Okay. It's a PS level type of hood?
Well, it's not a biosafety level, it's a chemical hood. So it doesn't flow in their face, but it takes it out, basically. And but in the production, I don't know about the factories, how they are, what cautions they are taking. I'm not aware if they take any precautions for the workers. They don't produce in mass yet, except for the multi-wall carbon nanotubes, but the multi-wall has been shown not to be that toxic to humans. So right now is not a lot of, there are not a lot of issues related to that. The single wall has been shown to be extremely toxic to humans, extremely, in the sense that it has kind of the asbestos effect, because it's very thin and it can puncture, and but it's not mass produced. And I know that, for example, in California, I used to buy from a company the single wall carbon nanotubes, and I think California vanished. Because when I emailed them to ask, they said, we are not producing anymore due to safety. But in other parts of US, they're still being produced. Now, the graphene and graphene oxide, it's really new. People have been starting to study really well. I don't know, when I started like 3.5 years ago, but before that, you don't see many papers, especially related to toxicity. You see a few papers more in the human side, but the human side, they didn't see a lot of toxicity for that, more in the microbial side. That's why I'm more interested in the effect in the environment. So wouldn't be an issue in the health side for the carbon-based nanomaterials. But, and one thing that we noticed is that whenever you combine nanomaterials with polymers, we have been noticing that it reduces the toxicity of the nanomaterials. So that's it's a big plus.
Thank you. Thanks.
Yes?
In the first part of your talk, you mentioned two ways that nanoparticles could damage a cell.
Mm-hmm.
Do we have an understanding of the physical processes involved in that? Is it mechanical indentation? Is it diffusion across the membranes, across the cell walls? Or?
I have my theory. I can share with you. We have seen some pictures of cells getting nanomaterials getting inside the cell. Okay, the exactly mechanism. My theory is because, especially the single wall carbon nanotubes and the graphene, they are hydrophobic. Okay, extremely hydrophobic, and the cell wall is made of lipids in most cases, which is also hydrophobic. So you can have a better partitioning inside the cell because of that. Does it make sense? But there is no proof. This is my opinion, based on what I know about cells and how nanomaterials also have...
But then could one design nanoparticles that are nicer to the cells? Or?
I guess, yeah. I mean, graphene oxide is nicer, and it has all the... because it's more hydrophilic. It likes more staying in the water than actually in the membrane. So, but still, it's still getting inside the cell because you don't have completely functionalization. So it finds its way inside the cell as well. So, and combining with polymers, making modifications, the beauty of having all these functional groups, COH, OH, is that you can bind stuff to it and make it less accessible to the cells. The same way that people change it to actually get it more accessible to the cells for drug delivery as well. So there are both ways that you can go basically with these nanomaterials.
So if we're using PVK and other polymers, what do we think the effect of that load on a wastewater treatment facility would be?
I'm investigating that. I am actually. I have a student that she started, because we started with the pure material, but I mean, those are advanced material. And in fact, what we know is that people are less and less likely to use the pure nanomaterial, but more combined with polymers. So we are trying to see what kind of polymers would be more interesting and what would be the effect to the microbial population if it was to go to the wastewater. Because I think it would change completely the properties because of all the chemistry in the water, organic matter and everything, and the polymer also will have some synergistic effect with the nanomaterial. So yeah, we are moving towards these advanced materials actually right now. Yes. Any more questions? Yes.
I think I'm missing the first part of your talk. What is the primary exposure pathway?
Of the nanomaterials?
Yeah. Are you talking about inhalation, or this is water?
It's water.
Water-based, okay.
I mean, you can have inhalation.
But that's more the occupational health.
Yes, that's what he left.
I've seen his
I was like, yeah, he left. Yeah, exactly.
Okay.
I mean, the professor from Brown University, he's looking at the air. How it gets and how it affects the respiratory system in humans.
Mm-hmm.
He has a collaborative project with some people from medical school on that.
Mm-hmm.
And I'm dealing the dirty part, wastewater, and soil as well, landfills. Have also some work on that. Yes.
This is a little outside the scope, so it's okay if you don't know the answer to this. But it seems like there's been some research in this area that looks at the effect of the photodegradation of plastics on microbial communities. And I'm wondering, are they affecting microbes using the same pathways, or is the issue with nanomaterials just their sheer small size? I don't know if you're familiar with that research or not.
Yes, I am. You were saying like photocatalysis, right?
Yeah,
degradation.
plastics. Yeah.
Yeah, I've seen such type of work, and it does happen with a lot of nanomaterials that you shine light, and then you have all these reactive oxygen species and degradation related to. I'm not investigating that because I'm a microbiologist. That would be more for a chemist type of person. But I mean, that would be really a nice combination, and I've always wanted to actually get a partnership with somebody that would do the chemical part of it, because that would definitely complement my research.
Mm-hmm.
Absolutely. And I see that point, yes, of degradation. I have one project that I submitted with a chemist on that, but it was for more titanium dioxide, not really for carbon-based nanomaterials. Actually, kind of. It's titanium dioxide coupled to graphene oxide, because you can actually combine, you can actually functionalize with titanium dioxide. Because titanium dioxide does this photocatalysis for degradation, chemical degradation. Graphene oxide, I'm not really aware of really anything that would really break down chemically. I haven't investigated and I am not aware of anybody that has done that. I've seen a lot with titanium dioxide and other zinc oxides and more the metallic nanoparticles, because they are extremely reactive with solar energy. They get activated. Yes.
What made you interested in environmental engineering and nanomaterials and all that fun stuff?
Oh my, that's a very good question. You know, let's see how I'm going to answer that. Everything started with my parents telling me like, biology? What the heck? Why don't you go to medical school? You have grades for that, you could go to medical school. And I was like, yeah, no. No, I like people, but I like to help people in a different way. And I was like, okay, how I can help people in a different way? Well, the environment is one way, because if we affect the environment, we affect a lot of people. So, and I had a fabulous teacher in environmental microbiology that I started working in her lab, and then I was like, wow, that's awesome! And I fell in love. And then I was like, medical? Nah. Environmental? Yeah! Big yes, because microbes, oh my gosh, the more I learn about them, what they can do, I'm like, they are amazing. They can do so many things, and I was like, okay, environmental microbiology, that's my call. And nanomaterials, that was my postdoc advisor. When I did my PhD, I worked with microorganisms and I saw how awesome they were. And then I did my postdoc with a professor that he worked with nanomaterials. And when I started to see what nanomaterials could do, I was like, wow, it's as awesome as microbes. So why not combine both? That's why, hence, my main goal is combining biotechnology with nanotechnology for my research. That's how it happened. And yeah, I'm extremely a passionate person. When I fall in love about something, forget it. Even my husband tells me that he's in third plan. First my research, my kids, my bacteria in the lab, then my son, and then my husband. He's like, I'm like, no, not quite true to say that. Come on. My son goes first. Anyhow. Yeah, but sometimes my son and my research is kind of anyhow. But don't say that to my husband. Well, any other questions?
All right, thank you very much.